This is chapter 21, dealing with rickettsias, chlamydia, spirochetes, vibrios. We're looking at microorganisms that are spirochetes. They have a microscopic morphology that consists of an endoflagella, typically two of them, one on either side of the longitudinal axis of the microorganism. They're spiral in morphology, and their motility, the ability that they use to move from one point to another, is as a drill, something that literally spirals in order to traverse the environment. This ability for them to spiral also allows them to enter mucosal linings and allows them an opportunity to move into areas that typically other motile microorganisms are not able to enter. So we start with chlamydias. Okay, the reason that we initiate with this particular pathogen is because it is the most common reportable sexually transmitted disease in the United States. So chances are that if you're in a general clinical environment, you will run into plenty of patients that are dealing with this infection. Chlamydia is interesting. It goes through a dichotomous life cycle. One is as an elementary body, and one is as a reticulate body. Okay, if you look at the picture here, here's the elementary body when the organism is outside of the cell. And the reason this is significant is because the elementary body does not have metabolism. It has attachment proteins, but it does not have a metabolism. So this organism is not susceptible to antibiotics while it's outside of the cell, because antibiotics function by being taken up by the organism, and again, that's something that's not taking place. So the elementary body is in a form of stasis. Again, it does have attachment protein, so it does have the ability to enter a host. Once it's inside the host, then that elementary body is turned into a reticulate body. So that means that the metabolism of the organism starts to manifest itself once it's inside the host. Once inside the host, in a phagosome that inhibits, it has an antiphagocytic factor where it's inside the phagosome, but the phagosome does not come together with the lysosome. So that means that this reticulate body can multiply in the phagosome inside the cell, and then in preparation for exiting the host, it goes from a reticulate body back to an elementary body, ceasing its metabolic function and exiting the host in order to traverse to a new frontier. Okay, so that's the cycle of the chlamydia. It can cause cell destruction. It can also cause the destruction of capillaries. So consequently, there is a red rash and inflammation associated with this infection. Okay, one of the main manifestations is called lymphogranulosa venerum, and it consists of genital lesions followed by buboes. What's a bubo? A bubo is when the adaptive immune system, in its effort to reconcile this infection, takes the chlamydia and it goes into the inguinal lymph nodes in order to carry out antigen presentation. The inguinal lymph nodes become saturated with this infection, and they start to balloon. Okay, that ballooning is called a bubo. if the bubo continues to mature and it ruptures, okay, then it can become a purulent draining sore. Okay. Interestingly, in many women, this infection doesn't have symptoms. Okay, and this is not a desirable event because if it goes untreated, as it may, then it doesn't mean that it isn't causing infection and damage, and it can eventually lead to pelvic inflammatory disease. And maybe some of you know that that can eventually lead to a woman becoming barren. Okay, so this is a trivial, is a relatively trivial infection that can become very problematic if it goes untreated, or what's the word I'm looking for? If it goes without detection. So there's a small example of a bubo in the inguinal area. Typically, they're even more inflamed than that. Okay. If we go to chlamydia trachomatis, now we're talking about problems that it causes with the eye. Okay. So particularly in neonatal events, when a child is being born, this can be passed to the eyes of the child. When the infection takes hold, it inflames the inside of the eyelid, and it causes it to turn inside out. In doing so, the eyelid causes pressure on the cornea of the child, and that pressure can lead to corneal scarring. So again, this is something that if the mother is known to have this infection, it needs to be treated prophylactically in the child to prevent blindness. So as an example of a poor child, you can see the oozing surface and the whitish infection under the eyelid, and the subsequent turning of the eyelid inside out. Okay. There's a beautiful immunofluorescent picture of chlamydia. As we've said before, this is diagnostic. Beautiful. Okay. And now we're switching to spirochetes. Okay. So again, as I said before, tightly coiled, helically shaped bacteria with endoflagella, typically 2 longitudinal flagella on either side. Consequently, they move in a corkscrew fashion. And why is this important? Because it allows the spirochete to burrow into the host tissue. It allows the spirochete to go into the mucosa and then into the epithelial lining, where other motile organisms might not be able to do this. The main genera of spirochetes, Treponema, Borrelia, and Leptospira. So here's Treponema pallidum, the causative agent of syphilis. Okay. Typically, this is transmitted via only sexual contact, although there's some literature out there saying that the spirochete can actually live outside body fluids for about an hour or so. So interestingly, it is possible to, hypothetically at least, transfer from one person to another via fomites. Again, endemic in sex workers, homosexual men, and people that share needles in illegal drug use. So this is a slide showing the spirochete. You can see that is very tightly, a very tightly wound helical pattern. Okay, and this is probably being grown in culture. If you're to see this density in in an organism, the organism would probably be dead. Here's the incidence of the of syphilis. You can see that we're looking at predominantly south west and southeast, okay, and mostly cleared in the central United States. The incidence has fallen over the years, but there has, as you can see here, there's been a slight increase lately, and we covered this in one of our summaries, addressing the fact that there's certain communities and where the stigma of having these sexually transmitted diseases prevent people from going to a physician and getting this treated. So that may be one of the main reasons that's attributing the late onset of this increase. Okay, it's important to know that syphilis, or Treponema pallidum pallidum, has 4 phases. Okay, that's a primary, secondary, latent, and tertiary. The primary phase of syphilis is what's called a canker, and this is an example of that. It is typically painless, and again, in women, that canker would manifest itself on the cervix, and the probability of it going unnoticed is actually pretty good. So, in about 1/3 of patients, this canker would manifest itself. Okay, and then it would heal itself after, it would resolve itself after a few weeks, and then in about 1/3 of the patients, we would go into secondary phase, which is what we call a widespread rash throughout the body, including the palms of the hands and the bottom of the feet, the plantar feet. So this is very systematic. It affects every aspect of the body, every part of the body, and even the brain. Okay. And, so 1/3 canker, 1/3 widespread rash, and then we go into what's called latent. That latent phase could be months, it could be years. Sometimes the latent period has been as many as 30 years. Okay, and after that latent period, we go into the tertiary, which is the gamma. Okay, in this phase, there is no spirochetes left. What has happened is that the spirochetes have changed the surface antigens of some part of the body, or many parts of the body. And those parts of the bodies are no longer being recognized by the immune system of the individual, and the individual launches an autoimmune response against those parts, and you end up with these so-called gamma that can manifest anywhere in the body. Here, this lucky person got it in the middle of their forehead, but it can be in any part of the body. It can be in any tissue. It's a rubbery, non-healing lesion that's being caused. The cells are being destroyed by the person's own immune system. And except for management, there's no real treatment because there's no spirochetes to kill. Okay, so you can give these person antibiotics, nothing's going to change. The only thing they can do is be careful that a secondary opportunistic infection doesn't take hold. Okay, treatment, interestingly, very simple, penicillin. Okay, again, ineffective against tertiary syphilis, and again, the reason for that is because there's no spirochetes at that time. Okay, now we're switching to Borrelia. With Borrelia, the one that we're going to look at is Lyme disease. Okay, and Lyme disease. Let me go one more here. So Lyme disease was discovered in the northeast United States. And the reason for this is because there was a higher than average incidence of arthritis in young children. Okay. So as we did the research to find out what was the source of this arthritis, we became aware that there was a causative agent, and that happened to be Lyme disease or Borrelia. Okay. So this is something that can manifest upon getting the infection. Again, this is an autoimmune effect, and consequently, it can last. It can last the rest of the lifetime of the individual. Okay, so this is interesting. Borrelia doesn't have any iron-containing cytochromes. So most organisms that we've talked about require iron, or they require sulfur, or they require copper in order to build the electron transport chain. Borrelia does not. Consequently, it would be it would carry out gamma hemolysis in blood agar. All of these require a vector, in other words, a delivery system. And here that delivery system is the hard tick known as Isodes. Okay, so being bitten by this vector is a necessary precursor to getting this disease. You can see the spirocheted Borrelia among red blood cells in this light microscope slide. And the life cycle is interesting. There's unaffected eggs that are laid on an animal. Those hatch into larvae. Those then jump onto plants and then subsequently into rodents or birds, where they become infected with the Borrelia. Okay. And then they end up growing and subsequently jumping onto other animals, including ourselves, biting us and transferring that infection, that Borrelia, onto us. And here we're showing that, what do you call it? The bullseye rash. Okay. So that's a very good, that looks very good. That's a very good example of the so-called bullseye rash. It can cause a lot of problems, as you can see. Neurological symptoms, cardiac dysfunction, severe arthritis. What's going on? Well, the spirochete can be systematic. As we keep on saying, it insists on changing surface antigens wherever it manifests itself. And that change of surface antigens causes the body to not recognize its own tissues. And so an autoimmune effects are being launched, okay? If that happens in the brain, then neurological symptoms manifest themselves. If that happens in the heart, cardiac dysfunction manifests itself. If that happens in the joints, arthritis manifests itself. And so this is a subsequent effect of the Borrelia. The Borrelia itself isn't doing this. It is the modification of antigens by Borrelia that is causing these autoimmune effects to manifest themselves. Here we're showing the increasing incidence of Borrelia in the United States. And you can see that it's happening predominantly in the northeast corner. Okay, why is this happening? Well, don't trust me, but global warming is causing all these emerging diseases that are typically understood to be tropical to move further and further north, and consequently to have the incidence increase more and more and more. So as the temperature increases, the incidence of this disease increases and moves further north. Let's see. Switching to Vibrio. Vibrio is interesting. I love playing with Vibrio. I have a tube of Vibrio fischeri here because it's a bioluminescent microorganism. So these are things that I find very interesting. Vibrio cholerae is the causative agent of cholera. So historically, this is a big problem. We've had probably millions of people die from cholera. Um, we have had recent pandemics of cholera. Most of them are in South America and some in Mexico, and lately even in Puerto Rico, because of the hurricane has mixed water, potable water with regular water, allowing this Vibrio cholerae to manifest itself where it typically isn't. Uh, there's uh, there's about 20 different species of Vibrio cholerae. Only one of them is known to be pathogenic. And what is significant about it is that this particular species can live in fresh water. Okay, it is the only one that can do that. And so this fresh water becomes potable water, water that we drink, and that's the one that we end up consuming, and that's what ends up causing problems. Okay, so pathogenic gram-negative Vibrio, again, one of the main significant traits is the fact that it is the only strain that can survive in fresh water. All the other ones require salt water. Okay, and what causes it so pathogenic? The fact that it produces a very powerful exotoxin called cholera toxin. Okay, and this cholera toxin, what it does is it causes a severe loss of fluids and electrolytes via the large intestine. Okay, so here's the epithelia of the large intestine. Here's a lumen. Here's a cholera toxin. The cholera toxin has attachment proteins that have an affinity for the microvilli of the epithelia. Once it attaches to the microvilli, it cleaves itself, allowing a subunit called A1 to enter the epithelial cell. Once inside the epithelial cell, A1 activates an enzyme inside the cell called adenylate cyclase. Okay, so I talked to this about this to my face-to-face class yesterday. It looked like nobody knew what I was talking about. Adenylate cyclase is an enzyme that is common in as a second in producing cyclic AMP that functions as a secondary messenger in many of the cascades in our body. Okay, so you can find it in the retina of the eye. You can find it in blood clotting, anywhere where you need a multiplicative event, where you have a multiplicative cascade. You're likely to find this enzyme whose sole purpose is to make the secondary messenger known as cyclic AMP. So as the adenylate cyclase gets produced, and this cyclic AMP gets synthesized from adenosine triphosphate, this cyclic AMP initiates the function of sodium and chlorine pumps that pump sodium and chlorine out of the epithelia. It makes the lumen of the intestine hypertonic relative to the intestinal lining, and consequently, water follows it. Okay, so this is the problem. You're getting rid of sodium, you're getting rid of chlorine. These are essential electrolytes, getting rid of water. And so if you have somebody in this condition, this is an immediate medical emergency that needs to be resolved. This person needs to get a lot of fluids, normal saline, wide bore IVs. Otherwise, their volemic volume is going to decrease very quickly, and they're going to enter anaphylactic shock and possibly pass away. So all this needs to be undermined by the introduction of copious amounts of normal saline in order to maintain electrolyte balance and water and hydration in the body. Okay. Interestingly, the author and other sources I've read say that the use of antibiotics at this time really is not useful for a couple of reasons. Number one, the antibiotics are never going to reach therapeutic level because they're being eliminated in the large intestine. Number two, the pathology is not being caused by the Vibrio. The Vibrio has probably been killed already. The pathology is being carried out by a toxin, and this toxin is not susceptible to antibiotics. So this becomes what's called a self-limiting event, and that means that sooner or later, this cholera toxin is going to lose its potency, and the patient is going to be able to resolve itself as long as they are maintained hydrated. Changing to jejuni, okay? So this is a different Vibrio. What is significant about it? It is the most common cause of gastroenteritis in the United States. Look at this, isn't this interesting? Poultry is the most common source of the infection. So before we said that poultry is the most common source of Salmonella typhimurium. Now we're saying that poultry is also the most common source of jejuni, which again causes gastroenteritis. And again, this creates a toxin that produces bloody and frequent diarrhea, something similar to Vibrio cholerae. Okay, again, self-limiting. As long as you keep the patient hydrated and maintain electrolyte balance, this will resolve itself in time. Here's pylori. Why are we talking about Helicobacter pylori? Well, for one thing, we have deduced that it causes gastritis, it causes peptic ulcers, and we believe it causes stomach cancer. So that's important to know. How does this happen? The fact that the spirochete pylori has the ability to enter the mucosa of the stomach. Okay, so that's one thing. The way that it moves, that motility, that spirochete motility allows it to burrow itself into the mucosal lining, but it also has adhesins that allow it to attach to the gastric lining. It also has enzymes that neutralize hydrochloric acid, and it even induces the production of bicarbonate in order to neutralize hydrochloric acid. So this is what it looks like. Okay, here's a pylori being able to traverse the mucosal lining of the stomach. Okay, as it produces enzymes to hydrolyze the mucosa and to neutralize the hydrochloric acid. It eats away at the mucosal lining of the stomach, and then it has the ability to destroy the epithelial cells, allowing it to traverse the stomach lining and enter the circulatory system. Obviously a problem, because you and I know that this lumen of the stomach is full of microorganisms, hydrochloric acid, chyme, a whole lot of things that should not be entering the circulatory system. So bleeding ulcers are a sincere problem and can be life-threatening. And that's the end to this chapter. I hope you enjoyed it, and I'll see you very soon.